Hello, my name is A.S. Ribeiro. I'm the principal investigator of the Laboratory of Biosystem Dynamics, which is a research group of the Faculty of Medicine and Health Technology of Tampere University. We are located in Arvo, where we have both our laboratories as well as our office. Today I'm here to present a paper that we recently published in the Journal of Nucleic Acids Research, which is about the transcription factor network of E. coli during global perturbations. I would like to start by thanking our funding agencies, the Jano and Ato Zerko Foundation, who has supported the project, without which it would not have been possible. I would also like to thank the Finnish Academy of Science and Letters, the Faculty of Medicine and Health Technology, the Finnish National Agency for Education, and the Finnish Cultural Foundation, all of which have supported our PhD students who have carried out a lot of the work. Moreover, I'd like to thank our collaborators, both in Finland as well as already outside of Finland, several of which were in the past PhD students of our Laboratory of Biosystem Dynamics, as well as researchers from other labs. Thank you all. For hundreds of millions of years, all living organisms, including bacteria, have passed on to their offspring the information of what they have learned during their life through the molecule called DNA. DNA is composed of genes that in some cases code only for RNAs, but in most cases code for RNAs from which proteins can be produced. Those proteins, some of them can carry out specific tasks. Other of them are transcription factors, which means that they can regulate other genes. Throughout evolution, they have formed networks. Thereby, nowadays, organisms such as Escherichia coli have a few thousand genes, and between them there's a few thousand interactions made possible by those transcription factors, as well as by other means. At any point in time, cells are being bombarded by information, and they have to process information. For that purpose, they have evolved these networks. These networks are capable of most likely multi-scale, semi-parallel information processing of these external signals, and at the same time, they are most likely significantly robust to spurious fluctuations. It is this that allows them to produce decisions that are robust and that are capable of, for example, timely activate very specific responses to very specific signals. The responses are produced by mechanisms that, in the case of signals that target very specific genes, for example, substrate availability, are relatively well understood. The substance comes in, it, for example, represses a repressor that is constantly expressed, and that repressor is itself repressed, which allows the genes to express in order to, for example, create or activate the mechanisms by which the substrate is consumed. But there are other perturbations that we know that cause genome-wide stresses. For example, a temperature shift most likely affects hundreds of genes, and we do have solid evidence of that. They affect not only a specific variable of the network, they affect multiple variables, multiple rate constants throughout the genome. And understanding how gene networks process these signals is yet an enormous challenge. But this is necessary if we want to understand how organisms make decisions, particularly when they're subject to environmental shifts and other life-threatening stresses. We know that individual genes are responsive to signals. We also know that genes communicate with other genes via, for example, transcription factor regulation. What we don't yet know, or we have significant difficulty in proving, is are there some decisions that are made not by individual genes, but by the gene network? For example, decisions that are influenced rather than by a specific gene or a specific transcription factor, the decisions are influenced by global properties of the transcription factor network. For example, by the mean number of transcription factors per gene, or for example, by the global clustering coefficient of the transcription factor network. It is that we set out to answer here, is whether, for example, short and long-term responsive genes, do they communicate via the transcription factor network, or if any of these two gene cohorts is influenced when subject to genome-wide stress, is actually defined or influenced by the transcription factor network and its global topological features, as well as logical. So more precisely, this is what we set out to investigate. We know that gene regulatory networks produce specific, repeatable, chronologically ordered responses to genome-wide stresses, such as temperature or media shifts. Do transcription factor networks play a role in this capacity? Or is this mostly driven only by individual genes, individual transcription factors, or very small subcircuits? For this, we considered the standard model of transcription regulation. There's a promoter which controls gene expression. Then there's a transcription factor which regulates that promoter and thereby gene expression. And RNAP is some sort of an engine that drives the system, where it binds to the promoter region, and based on the transcription factor presence or not, it can transcribe the gene at a certain rate constant K. RNA is the product of these reactions, and from the RNA, proteins are produced. We set out by producing a shift in RNA concentration that would quickly initiate a global perturbation by changing the rate at which many genes are being transcribed. And then to study the responses, we conducted RNA-seq, flow cytometry, microscopy, some Western blot, and spectrophotometer. That was the techniques that we used. But one key element of our study is that we obtained a very concise and complete information about the transcription factor network of the organism that we study, Escherichia coli. So we went to RegulonDB and we obtained the transcription factors, what's the input, what's the output gene, and moreover, what is the regulatory effect of that transcription factor on that output gene. Is it an activator transcription factor, or is it a repressor transcription factor? And it's based on regulatory effect, as well as on the number of inputs on a specific gene, that we produce a hypothesis of what would be the response of that gene to the perturbation. And based on that, what would be the global response as well to the perturbation. To that effect, here's what we expect that will happen once the perturbation is caused. There will be an RNA polymerase change. And once this occurs, we observed that it took around 60 minutes. Following a certain amount of time, approximately 60 more minutes, the RNA polymerase has already changed, and the RNAs of the first set of genes, which we call the short-term response, have already changed as well. We expect most of them to be reduced in numbers because the RNA polymerase has lowered in concentration. But as these RNAs are reduced in numbers, so are the proteins that they code for. And some of those proteins are activators, some of the proteins are repressors. In the case of the proteins being activators, the output gene will also have their RNA numbers reduced. But in the case of being a repressor, the output genes will actually have their RNA numbers increased. And this midterm response, unlike the short-term response, which is probably only going to depend on the change in RNA polymerase concentration, the midterm response will already depend as well as on the transcription factors changes in addition to the RNA polymerase changes. Here we did not study the long-term response because we would not be able at this stage of processing the influential variables because there may be too many influential variables for us to decode the signal. And here there is the experimental results of our studies. In figure A, we show you the RNA polymerases quantities in individual cells on average, has shown by spectrophotometry of cells carrying an RNAP which is tagged with GFP. We did two perturbations, we call it weak, medium, and strong, which are corresponding to weak, medium, and strong change in media richness. You can see the quickness of the response. After 45 minutes in the media, the cells are already starting to exhibit the changes in RNA polymerase levels, and after 180 minutes they stabilize in those levels. So on the other hand, we also saw it from the point of view of concentration by observing microscopy data, and you can see there's the control, which we call 1x, and there's the 0.75x, which is the weak perturbation, the 0.5x, which is the medium perturbation, and the 0.25x, which is the strongest perturbation. And as you can observe, not only the total amounts change, but the concentrations of RNA polymerase in the cells change. That, based on the chemical reaction model below, should change the overall propensity of the reaction promoter transcription factor and RNA polymerase, which leads to the production of RNA. Then we conducted the genome-wide measurements. For that, we performed RNA-seq. You can see there the average absolute log-fold changes in RNA levels as measured by RNA-seq for each one of the three perturbations. We see from weak to medium to strong, relatively speaking, changes in absolute log-fold changes per gene. So as you can see, there's significant perturbations. In the final graph, what we observed is the number of genes that were perturbed in a statistical sense as observed and analyzed by RNA-seq. So you can see that, for example, there's a significant correlation, let's say, between the experiments, that is, genes that are perturbed in the strong perturbations and in the weak perturbations and in medium perturbations are correlated. It's not just random genes being perturbed. So, for example, at 0.75x, only 19 genes that were perturbed significantly were not perturbed in the 0.5 and the 0.25. But the majority, the 141 that were perturbed for weak perturbations, were also perturbed in medium and strong perturbations. You can also see a very strong increase in total number of genes perturbed. For 0.75x, more or less 200 genes were perturbed. For 0.5x is already almost 1,000 genes, and for 0.25x, approximately 2,000 genes were already perturbed. So this can be considered a genome-wide perturbation in an organism that has approximately 4,500 genes. Having measured the changes in gene expression levels from the genome-wide perturbation, we next studied the transcription factor network topology. As said, we obtained this topology from RegulonDB. All the transcription factors known, their inputs, their outputs, as well as their regulatory effect. But here we're just focusing on the topology. The global topology can be said to have the following properties. First, you can see that the in-degree distribution follows well a power law distribution with the parameters shown in the figure. And then we compared the E. coli network also with what are the average properties of Arnairdos and Renier random topology, as well as with a random network with a scale-free topology. So we recorded 4,000 nodes, that is genes, in E. coli. So we set the models of the Erdős-Röntgen Régnier and the scale-free topology with the same number of genes. The number of transcription factors, that is edges, was 4,471, and we imposed the same in our null models. And then you can see the result. In E. coli, we have 0.06 clustering coefficient. On the other hand, in an Erdős-Régnier and in a scale-free random topologies, we have a much smaller, 100 times smaller clustering coefficient. This clearly shows that the evolution of E. coli, the transcription factor network of E. coli, has significantly increased the cluster coefficient from what would be expected by random chance. Also interestingly different are the average path length. It's clearly of the scale-free type rather than of a random Erdős and Régnier topology. But there's also a significant number of isolated genes when compared to the random models. 2,300, when in the random models there would be around 400. And I believe this also has a lot to do with the clustering coefficient. For you to have such a clustering coefficient, you probe such a higher clustering coefficient than the random models, you're going to pay, quotes and quotes, in the sense that more nodes will have to be isolated given a fixed number of edges. We also observed a very significant high number of self loops of genes that regulate themselves in the natural system, E. coli, when compared to the random models. For information to propagate, it's essential not only the existence of an interaction, but also the regulatory effect of that interaction. Some transcription factors are repressors, some transcription factors are activators. That has been already measured or identified in the case of E. coli, of many interactions in E. coli, but it has not been identified in any other organism. Also, even in E. coli, we have only identified this to a certain level of simplicity. Like, for example, we know that transcription factor 1 is an inducer, but we don't know if that's still true when there's also transcription factor 2 present, and so on. So there's no clear identification of complex functions such as an OR function or a NOR function. So we assumed a simpler system than Boolean functions in what concerns the logic of the transcription factor network. We call it an approximate logic. And the way we assumed it is the following. Imagine the example exhibited in figure C, where we have 3 transcription factors, 1, 2, and 3, and 4. So the idea is something like this. If all 4 transcription factors are repressors, we assume a bias B of -4, in that they sum each other, -1, and so on. If they're all activators, +4. If 2 are activators and 2 are repressors, then they cancel each other out. Now, of course, we know that some transcription factors are strong, others are weak, and so assuming that they all have the same strength of 1 or -1 is simplistic. And that's why we call it approximate. In any case, what we try to do is, based on the topology and on this approximate logic, where what matters is the overall bias of the set of transcription factors of a gene, we are going to observe whether this logic and topology can predict the dynamics that we reported from the RNA-seq data following the genome-wide perturbation. In summary, we have made a genome-wide perturbation with the RNA polymerase shift in concentration. We have obtained the topology and the approximate logic from Regulon DB, and our question is, can we predict the dynamics from the logic and the topology? We then went and confronted the logic and topology of the transcription factor network with the dynamics of the responses to the genome-wide perturbation caused by the shift in RNA polymerase. In order to verify if the topology and the logic have a genome-wide effect on the dynamics, we first compared whether genes with and without input transcription factors, so regulated by transcription factors and not regulated by transcription factors, had different response strengths to the perturbation. In figure A, we observe that they do respond differently. Approximately 2,300 genes are not regulated by transcription factors, and we observed 1,700 genes that are regulated by transcription factors, where KTF is the number of input transcription factors. Visibly, for the weak, medium, and strong perturbations, in all cases, genes with input transcription factors exhibit a stronger response, as observed by the log-fold change, the absolute log-fold change, to the genome-wide perturbation as measured by RNA-seq. This is a significantly strong difference and suggests or verifies that, in fact, transcription factors are influencing the response. So there is information, there is signal being propagated by transcription factors. Is that propagation actually correlated between inputs and outputs? And for that, we calculated whether there is a correlation between the log-fold change, the response strength of inputs, and the response strength of the outputs. As shown in figure B, the blue line is the null model, where we randomize who's an input and who's an output. Clearly, there's no correlation. In green is the maximum possible correlation that you could obtain if you were choosing who's the input and who's the output. And in red is the real, the measured correlation between input and output. The linear fit is clearly inclined in this scattered plot, and of course is statistically significantly different from the blue line, and thereby we conclude that with an R squared of 0.11. So we conclude that indeed the response strengths of inputs and outputs is correlated, again confirming that there's information propagation. So the transcription factor network is playing a role in the global response at the specific point in time after the perturbation. Interestingly, in graph C, we observed that the correlation between input and output decreases with the path length. So if gene A has a transcription factor to gene B, that's a path length of 1, and then from B to C, it's a path length of 1, but from A to C is a path length of 2. And we plotted for path lengths of 1, 2, and 3 in the transcription factor network, and you can see the correlation, the inclination of the scattered plot, of the linear fit to the scattered plot, gets smaller and smaller, and by a path length of 3, you can no longer distinguish from a null model of no correlation. And so indeed the signal did not propagate that long. Now there's a reason for that. It depends on when did we measure, because it takes time for RNAs to be produced and for proteins to be produced. And at the time of our measurements, we did not expect, based on known RNA protein production times and folding times and activation times of proteins, we do not expect the path length of 3 to be possible. So a propagation of path length of 3 to be possible. And so these results are very consistent with how long do we expect to take for a gene, once activated, to produce an RNA protein, and for that protein to then affect, if it's a transcription factor, its output, and then that output gene changing its RNA number. Finally, in graph we plotted the absolute log fold changes as a function of the number of inputs of a gene. So we have their cohorts of genes, and we plotted their log fold changes. For more detail, please see the paper. We shall focus now on the average result for all genes, dark blue. And visibly, in all cases, it's growing. The log fold change increases with the number of input transcription factors that the gene has. So if you have inputs, your response is stronger, and the more or the higher the number of inputs that you have, the stronger was your response on average. So again, a global parameter of the transcription factor network topology, the average number of inputs, influences the response strength of the genes. Originally, we hypothesized that the relevant parameter for the information propagation between inputs and outputs, or the relevant parameter for the log-fold changes of individual genes, is the bias of their set of input transcription factors. So if the signal is propagating throughout all genes and all transcription factors at a given point in time, for a certain number of inputs and outputs that nullifies each other, we should not have a significant propagation of information. But if they're all activators or if they're all repressors, the propagation should be very strong. The effect should be very strong in the output. And so instead of plotting the log-fold changes, absolute log-fold changes so the response strength has a function of the number of inputs, KTF, we instead plot it as a function of the bias in the inputs, which we call here B. And visibly, that cohort absolute response increases very significantly with the bias. Between no bias, there's a certain response, and a bias of 2, there's an increase of approximately 40% in the response strength. So we need to finally ask our question. So is it the number of inputs or is it the bias? But based on our hypothesis, it should be the bias. Following from before, we plotted the response strengths of genes, the average response strengths of genes as a function of the bias in the set of input transcription factors that controls them. We plotted that as a function of the response strength, and visibly in all cases there's an increase in the response with the bias of the inputs, and there's an increase also with the perturbation strength as expected. And next, in order to understand if it's the number of inputs or if it's the bias in that set of inputs, we plotted the number of the inputs against the bias of those set of inputs. And visibly, there's almost a perfect correlation between the two, which could be expected in the sense of that as the number of inputs increases, the bias that can be in them, in their overall regulatory effect should increase as well. So a set of 3 inputs should be able to cause a bigger bias than a single input. And thereby, we need to do more if we want to understand which of these actually is controlling the response strength on average. In order to test if it is the number of inputs or the bias in those inputs that controls the average response strength, we use the ensemble approach. That is, we generated from the empirical data random sets of gene cohorts as a function of their number inputs and the bias in those inputs. So first, we generated cohorts where the bias is always 1, while the number of inputs can grow. You see in graph B that the light blue is a straight horizontal line as a function of KTF in terms of the response strength. That is, for genes whose set of inputs has a bias of 1, increasing the number of inputs itself does not change the response strength. Similar, for genes whose set of inputs has a bias of 2, if that bias is fixed and the number of inputs increases, there is no increase in the response strength. Overall, what this result tells us is that changing the number of inputs does not alter the response strength if the bias of those inputs is fixed. On the other hand, it also tells us, you can see that those lines keep on going up. It tells us that if the bias increases, the response strength increases. In agreement, we did the opposite to validate this conclusion. That is, we make gene cohorts, we assemble gene cohorts with a fixed number of inputs, but with increasing bias. And visibly, you can see that for a specific number of inputs, if you increase the bias, you increase the average response strength. And so we conclude by using this ensemble approach that randomly generates sets of gene cohorts with specific features, we conclude that it's the bias that controls the response strengths of the genes, not the number of inputs of those genes. In summary, we subjected the gene regulatory network of E. coli to a genome-wide stress caused by changes in RNA polymerase concentrations. We were able to obtain a very detailed model of the gene regulatory network of E. coli because it is very well mapped in terms of the number of inputs, where do those inputs connect, which genes are being connected, and whether the input transcription factor is a repressor or an activator. Based on that, we were able to establish the set of input transcription factors of each gene, as well as the bias that they may cause due to the differences in numbers of activating and repressor input transcription factors. We found a very interesting result in that as the number of input transcription factors and thereby on average the bias, as that bias increases, so does the average responses to this genome-wide stress. To the best of our knowledge, this is the first biological phenomena that is behavior of the gene network that has been empirically linked to the global topology of a gene network, and in this case being the average bias or the distribution of bias in input transcription factor regulatory effects. It is interesting that in the past, the models of gene networks, perhaps the most used has been Boolean networks, which basically considers the interactions, the input transcription factors, and then it assumes that they have a positive or negative role, but then it allows for complex Boolean functions from sets of inputs. This data suggests that it's slightly simpler than that. We have to consider all the inputs, we have to consider the distributions of inputs numbers on individual genes, but then we can create a more simple variable rather than all possible Boolean functions. It's that bias in the regulatory effect, activation or repression in the set of inputs that controls the global dynamics. We're thus very interested in now studying in models and then of course in empirical data. We're very interested to study what is the global dynamics of models that would take these results into consideration. And of course, we want to study if our results apply to other perturbations in E. coli, observe whether, provided that we have a better knowledge of gene networks, observe whether the same phenomena can be visualized in other bacteria. And interestingly, we have found that not all global topological features of E. coli's transcription factor network are similar to a random network. And so we hypothesize that they may be significantly influential also in the genome-wide behavior of the network. Perhaps other perturbations targeting specific regions of the network might allows us to study this phenomena. And overall, we believe that the present paper can be used already to construct a new genome-wide dynamical model of gene networks, which we can then use to predict and then test the global behaviors of this network. I thank you all for your attention, and please don't hesitate to contact us if you have questions, suggestions, or just want to discuss the work itself. Thank you very much.